Great. So welcome to Genevieve Clutton. Genevieve is a research associate at the University of North Carolina at Chapel Hill. She received her DPhil at University of Oxford, where she studied the role of T-cell-derived IL-10 in HIV-1 pathogenesis. In 2014, she moved to Chapel Hill, initially joining the university's HIV cure program and elucidating the effects of putative HIV latency-reversing agents on HIV-specific T cells. Subsequently, she then broadened her focus to study T cell function and metabolism across a range of human diseases. And her research focuses on cell-intrinsic factors that contribute to T cell dysfunction in chronic viral infection and cancer. And the title of the talk is CD3 downregulation identifies high-avidity human CD8 T cells. So thank you very much, Genevieve. Over to you.
Thank you, Jeremy, and good morning, everybody, or good afternoon or good evening, depending on your time zone. And thank you to ProImmune for the invitation to present some of our recent work on the use of CD3 downregulation to identify high-avidity human CD8 T cells. So I'm fairly confident that everyone on this call is aware that CD8 T cells recognize infected or malignant cells that are presenting peptide on their surface, typically via MHC class 1. And the CD8 T cell recognizes this via its T cell receptor, which is in complex with CD3 invariant chains, and the CD8 co-receptor. And as Jeremy actually alluded to earlier, following this interaction, the TCR complex and CD8 are downregulated from the cell surface. Now, in the human population, there is an enormous diversity of T cell receptors. And this is one of the factors that will affect the strength of the binding between the CD8 T cell and its target. And when we think about the strength of this binding, we can speak about affinity, which is simply the interaction between two proteins, ligand and receptor. We can talk about avidity, which is the sum of all the interactions and binding interactions between the CD8 T cell and its target. Or we can talk about functional avidity, which is essentially the sensitivity of the CD8 T cell to low concentrations of its cognate peptide. And so we have an example here where you can see that as we reduce the peptide dose that the T cell is exposed to, the lower avidity response is lost. We lose the ability to detect it, whereas we can detect a functional response by the higher avidity T cell down to a much lower peptide concentration. Now, when we think about T cell immunotherapies, knowing the avidity of the T cells that we are attempting to elicit is extremely important. Higher avidity T cells have been reported to promote more efficient tumor rejection and superior virus control. And in fact, recent work published in Science by the Mark Connor's lab suggested that ineffective HIV vaccines that have been tested clinically have elicited rather low avidity T cells. However, in certain contexts, high avidity cells may increase the autoreactivity risk, perhaps in cancer if a mutated self-epitope is being targeted. And it's also been suggested that high avidity cells in certain contexts could drive more rapid viral escape. So how do we measure affinity and avidity? Well, there are a number of methods, some of which are shown here. Some of these methods are rather low throughput and they may require specialist equipment that is perhaps not in the reach of a standard clinical laboratory. Staining with multimers, such as pentamers, is another approach that has been reported. Due to the incredible diversity of T cell receptors that we just discussed, for a clinical cohort, you would need probably a large number of multimers to cover all of the different responses that you might be interested in. Interestingly, however, it's previously been reported in T cell clones that higher avidity T cells downregulate the T cell receptor to a greater extent than lower avidity cells. And this, in a clinical context, is something a little bit more tractable, because instead of using antibodies against the highly variant T cell receptor, we can use antibodies that target the invariant CD3 chains that are part of that complex. And so then we can use one antibody for all the different patients in our cohort. However, for this actually to be useful clinically, this observation would need to be maintained in a clinical situation where we have patients of different ages, sexes, infection histories, etc. And so this was the question that we set out to address. So our approach was to use a cohort of HIV-infected and uninfected individuals. Our HIV-infected participants were durably suppressed on antiretroviral therapy. We had male and female participants across a broad range of ages, and the majority were also CMV co-infected. We used interferon gamma ELISpot to screen these participants for responses to 64 different individual epitopes from 3 viruses, CMV, EBV, and HIV, and we identified responses to 16 of these epitopes across the cohort from the 3 different viruses, with 8 different MHC class 1 restrictions. To examine CD3 and CD8 co-receptor expression on the surface of these cells, we then used pentamer staining ex vivo. We used intracellular cytokine staining to look at expression by cells that had been exposed to their cognate peptide. And to measure functional avidity, we used interferon gamma ELISpot with peptide titration. So first of all, we looked at CD3 and CD8 expression on the surface of virus-specific CD8 T cells ex vivo using proimmune pentamers. And so we compared the expression of CD3 and CD8 on the surface of our pentamer-positive cells to the expression on all of the other non-responding cells in the same culture, shown in this representative example here. Now, to allow us to compare measurements on the same scale between participants and experiments, we normalized the expression of CD3 and CD8 on the non-responding cells to 100%, and then compared the expression on the virus-specific cells to that threshold. So if a data point appears above the line, that means that the virus-specific cells expressed more CD3 and CD8 on their surface relative to the bulk population. If they're below the line, that means the expression was lower, and this will be repeated on subsequent slides. So hopefully you can see that ex vivo, virus-specific cells expressed similar or slightly lower levels of CD3 on their surface relative to the non-responsive population. Importantly, this may be partly a consequence of the pentamer binding itself. And their levels of CD8 were similar or perhaps slightly higher relative to the other cells in the culture. But importantly, we did not see that these virus-specific cells expressed very low levels of CD3 or CD8 ex vivo prior to stimulation. When we then looked at levels on the surface following antigen stimulation by intracellular cytokine staining, we saw extensive downregulation of CD3 and CD8 from the surface by responding cells, as we expected. And we defined responding cells as perforin low, CD107A positive, interferon gamma positive. This is the same participant from the previous slide. You can now see that the CD3 and CD8 levels are much lower compared with the bulk population. And we observed this across the cohort for CMV, EBV, and HIV-specific responses, although as you can see, the CMV-specific responses typically downregulated CD3 and CD8 to the greatest extent. Notably, cells responding to PHA, which does not engage the T cell receptor in the same manner, did not downregulated CD3 or CD8 to a limited extent, if at all. So our next question was whether we saw the association with avidity that had been reported in T cell clones. Here, as I mentioned, we used an interferon gamma ELISpot with peptide titration. This is an example from one participant where hopefully you can see that the CMV-specific response in blue was of the highest avidity as measured by area under the curve, and the HIV-specific response was of the lowest avidity. And this was recapitulated across the cohort. The CMV-specific cells tended to be of the highest avidity, in line with previous reports. Therefore, because we had seen this difference between viruses, when we were looking for associations, we used a partial correlation analysis to control for a possible effect of viral specificity. And even when we took virus into account, we saw strong associations between CD3 and CD8 downregulation and avidity. That is, the higher avidity CD8 T cells downregulated CD3 and CD8 from their surface following stimulation to a greater extent than the lower avidity cells. We were keen to develop mathematical models to describe the relationships that we had observed and also determine whether CD3 or CD8 was a more reliable predictor of functional avidity. And so here our statistician colleagues generated generalized linear models with log links. So the data that you see here are the same data as from the previous slide. It's just that the avidity has been log transformed to allow linear relationships to be described. So just focusing on CD3 or model 1 for a moment, they generated, to my non-statistician eyes, a very frightening looking equation. But essentially what this equation describes is that for every 1 unit increase in CD3 relative MFI after stimulation, avidity is predicted to decline by 1.4%, and the confidence intervals are shown here. Now you'll notice that the model has 3 different lines representing each virus. This is because the model predicts that absolute avidity at any given CD3 expression is partly dependent on virus. However, what you can hopefully also see is that these lines are parallel. And importantly, what that means is that regardless of the virus targeted, the relative change in avidity is predicted to be the same as CD3 increases. So what this means is that this model can be used for any pathogen if we're wanting to distinguish higher and lower avidity cells. They then generated a similar model for CD8, which I'm not showing here for time. And then they used the Bayesian information criteria to infer that the CD3 model was preferred as a more reliable predictor of CD3 than CD8 of avidity. So you could just use CD3 staining if you wanted to. So we demonstrated that this association between downregulation of CD3 and CD8 and functional avidity was maintained in a diverse human cohort with lots of demographic variables. But we wanted to ask if we still saw this relationship if we took every variable out of our system except for the affinity of the interaction between the TCR and its cognate peptide. And we wanted to look beyond viral infection to a cancer model. So here, working with our collaborators, we used human T cells that had been transduced with a single T cell receptor whose cognate antigen is shown here. It's a leukemia-associated antigen, and it binds the TCR with high affinity. We then used alanine substitution to generate a substitute peptide that was predicted to bind MHC with the same affinity, but the T cell receptor with lower affinity. We used MHC stabilization experiments on T2 cells to demonstrate that indeed the substitute peptide did bind MHC, and in fact with slightly higher affinity than the index. Yet when we looked at the T cell response, the response to the substitute peptide was of lower magnitude and it was of lower avidity, showing that the difference was due to the interaction between the peptide and the TCR. We then confirmed, looking just at the responding cells, that's the interferon gamma positive cells, that cells responding to the higher affinity index epitope downregulated CD3 and CD8 to a greater extent than those responding to the lower affinity substitute. Finally, we asked whether CD3 and CD8 downregulation could predict viral escape in HIV infection. And viral escape occurs when a mutation in a sequence recognized by a T cell, a viral sequence, reduces the ability of that T cell to detect the viral epitope. And we built on the work of a previous graduate student in the lab who had used viral sequencing and ELISpot to identify viral sequences that had escaped and not escaped from that individual circulating T cell response. So we used the same avidity ELISpot approach to confirm that while responses to the escape epitope were detectable, they were of substantially lower functional avidity than the responses to the non-escaped epitope. And consistent with all of our other observations, we saw that cells responding to the higher avidity index peptide downregulated CD3 and CD8 to a greater extent than those responding to the lower avidity escape peptide. To summarize, I hope we've demonstrated that downregulation of CD3 particularly and CD8 following antigen stimulation is highly correlated with functional avidity in primary human T cells, regardless of age, sex, the MHC restriction of the epitope, and regardless of the pathogen targeted. Mathematical modeling indicated that CD3 was the stronger predictor of functional avidity. And so this means that a simple flow cytometry assay, it could be as few as 5 colors, can be used to screen for high versus low avidity responses in a clinical context. A limitation is that with the sample size that we had, the model we generated does not provide a precise quantitative estimate of functional avidity based on a future value of CD3 or CD8 expression, but rather it allows us to distinguish higher and lower avidity CD8 T cell responses, even if they're from different individuals and regardless of the pathogen that we are interested in. I'll wrap up by acknowledging everyone in the lab and our collaborators who contributed to this work, our clinical colleagues who recruited the participants themselves who made all of this possible. I'd like to thank our funding sources, and I'll be very happy to take any questions. Thank you.
That's great. Thank you very much indeed, Genevieve. A really nice, clear presentation. It's fantastic. So we do have a first question from Etienne, who says, great work. What are the kinetics of CD3 and CD8 downregulation? When does reconstitution happen?
That's a really great question, which we did try to address in our intracellular experiments. In time course experiments, we observed that downregulation of CD3 and CD8 is already apparent by an hour post-stimulation and reaches its maximal level between 3 and 6 hours. The limitations of that approach are that, of course, earlier we don't really necessarily have a functional readout to look at. So there was some evidence that even at 30 minutes, we were seeing some downregulation, but we have relatively low numbers of responding cells at that point. And then in terms of the reconstitution of CD3 and CD8 at the cell surface, the issue with intracellular staining is that monensin and or Brufeldin A is added to the culture, which prevents trafficking of proteins through the ER and the Golgi to the surface, and therefore, depending on the method of cycling of CD3 and CD8 and whether newly synthesized CD3 and CD8 is coming to the surface, that treatment is going to block the recovery of the levels of CD3 and CD8. And so in our hands, we saw low expression out to 24 hours, but I really can't comment on what that means for the reconstitution, because the only reconstitution that we would see in that situation would be of CD3 and CD8 being recycled to the surface that had not transited through the Golgi. So I think our approach limited us in that respect. But certainly the downregulation is rapid, but I can't really comment based on our data about the recovery of those levels.
Great, thank you very much, Genevieve. Jorge, would you like to ask your question verbally? If not, no problem, I can ask it for you. He says, what can be the applicability of this downregulation in the clinic?
So what we envisage is if you are screening, let's say, a patient for T cells to expand ex vivo, for example, just to give one example, and in the context that you are interested in, you want your cells to be of as high an avidity as possible. The cell, you might expand your cells to a panel of peptides, and you want to know which of your responding cells, maybe you got 3 or 4 hits, is of the highest avidity. So using fairly simple flow that doesn't take a lot of cells, you can compare the expression of CD3 and CD8, if you like, on the cells that have responded to your hits, and the one that has downregulated CD3 to the greatest extent, our model predicts would be the highest avidity cell. Another potential approach would be vaccine testing. So you have a new candidate HIV vaccine, for example. Does it elicit cells of a higher avidity in your vaccinated participants than was seen with responses to previous vaccines that were not efficacious? So that's just a couple, but there would be numerous.
Great. Oh, thank you very much. Well, fascinating. I'd love to be able to spend more time talking about avidity of T cells. It's a really, really cool subject. So yeah, thank you for joining us, Genevieve. Really appreciate it. So thank you very much for speaking today.